Good afternoon. My name is Andrea Bustos. Today I will be presenting Dj1 expression and dopamine deregulation in HIV-infected antiretroviral therapy associated in neurodegeneration. So according to the World Health Organization, 3.7 million people around the world in 2020 are living with human immunodeficiency virus, which is also known as HIV-1. So one of the effectiveness in the US, the combination antiretroviral therapy, CART, also known as CART, drugs have prolonged the lifespan of people with HIV, aiding the patients to age with the virus. One of the consequences that is known with these patients is because they're going to live with the virus, they are also going to have to take the CART drugs and will have long-term effects of CART in the aging population, which is causing neuropsychiatric disorders, such as depression. So among the aging population, depression is prevailing, and clinical evidence has established the prevalence of depression among CART-treated patients. And one of the molecular mechanism of CART induces depression-related degeneration has not been established yet. So the objective of the study is to establish the correlation of Dj1 expression and CART drugs and HIV. So for the methods for differentiation of SY5Y of human neuroblastoma cell lines and CART. So first, cells were seeded SY5Y cell line, then were treated with retinoic acid. 72 hours were used for an incubation period, treated one more time with retinoic acid, 72-hour period was also given for another 72 hours. After the 72 hours were up, CART was used to treat the cells with dolutegravir, imetroxacitabine, and tenofovir at different concentrations, high, middle, and low. So after the cells were treated with CART, 72 hours were used for incubation. So once those 3 days were gone, it was available for the cells to be viewed for the morphological changes and to storage the pellet. So in order to storage the pellet, that would be useful for RNA extraction for PCR, as well as Western blot. So for the MTS proliferation assay, cell seeding of differentiated SY5Y, which was what I mentioned earlier, and as well as the SY5Y cell line. So those are not treated with retinoic acid. So they will be treated with CART drugs, which is the drugs I mentioned before, dolutegravir, imetroxacitabine, and tenofovir. That will be incubated for 72 hours. The addition of MTS reagent will be added once the 72 hours are up. Then there will be an hour incubation for the colorimetric reaction, and the plate will be read at an absorbance reading of 490 nanometers. So one of the key findings that we see here, and one of the comparisons that we have, is that we will be seeing the morphology of SY5Y and differentiated SY5Y after 72 hours post-treatment of CART. So for the control, in comparison with dolutegravir, imetroxacitabine, and tenofovir, we have a low concentration and a high concentration. So one of the changes that we start to see here is that in tenofovir, you start to see some cells changing compared to the control with a low concentration. However, at a high concentration, there is absolute death. They're not obvious, and the shape of the morphology has changed. As well as for the imetroxacitabine, but it's not a big comparison compared to tenofovir. Dolutegravir seems to have some morphological changes at low concentration as well as high. So for the morphology of differentiated SY5Y, you can compare them to the control, and for the low concentrations, it is obvious that dolutegravir starts to have some changes within the cell density. However, one of the key things to point out is that all three drugs has differentiated neurites. So you start to see that neuronal phenotype in each one. However, in dolutegravir, once it reaches at a high concentration at 72 hours, there is cell death. In amitriptyline, there is some morphological changes as well, but the key thing in tenofovir and dolutegravir is that you start noticing a big difference in these two concentrations, which are high. For the cell viability of differentiated SY5Y with CART 72-hour exposure, as I mentioned before, our drugs of interest are dolutegravir, amitriptyline, and tenofovir. One of the key things to notice here in comparison to the control is that once the drug concentration increases, the cell viability starts to decrease. And dolutegravir painted a great picture here with the different concentrations. With amitriptyline, you start to notice the differences in concentrations with the cell viability, which starts to decrease as well, and tenofovir as well. So once the concentration is low to high, you start to notice a difference compared to the control. So for the cell viability of SHSY5Y with CART 72-hour exposure, dolutegravir also starts to notice that there is a change in cell viability as it starts to increase in concentration. So for the amitriptyline and tenofovir, they both start to also decrease as the compared to the control as they increase in concentration. So in the results, one of the key things I wanted to mention was that the comparison of cell viability between differentiated SY5Y and SY5Y was to perform, to identify the differences in the levels of pathological morphology changes in neuronal phenotypes with the effects of CART for long exposure. In conclusion of this poster, the differentiated neuronal cell viability indicates pathological morphology changes after 72 hours of exposure at a high concentration compared to the control. So one of the key things that I also wanted to mention was that the morphology of neuronal cells treated with different concentrations significantly changed with an increase in concentration of CART for a long exposure of time. So one of the things that I did mention here was that the 20 and the 1.8 were the big indicators that once the concentration increases, the morphology changes. So future works characterizing the correlation between HIV and CART-mediated neurodegeneration will contribute to early detection of neurological disorders. And I want to thank NIH for the grant for allowing me to be able to continue my studies, and also Dr. Roy, Ms. Roy, Dr. Gill, and Dr. Rodrigo. Thank you all.